So this is a demo for Neu3CA-RT, a framework for real-time fMRI network analysis and neurofeedback. We have developed this toolbox here at our research group Neu3CA at the Eindhoven University of Technology. The basis for this toolbox is to provide neurofeedback to a subject inside an MRI scanner. And this is the experimental setup with which we do that. We have an MRI scanner from Philips, and we transfer functional fMRI data in real time to an external PC where we do real-time network analysis. The idea is to get a characterized brain activity pattern in real time, which we then map to a set of 50 behavioral profiles as defined by the BrainMap behavioral toolbox. These behavioral profiles are then much more intuitive for interpretation of brain activity and can be used as neurofeedback to the subject or for other applications. Our network analysis model is based on work done by researchers at the Medical Image Processing Lab at the EPFL and University of Geneva. Their ICAPS framework consists of a set of temporally and spatially overlapping brain networks that one can use to dynamically assemble known resting state networks. This is also conveniently correlated with the BrainMap behavioral profiles. This ICAPS framework is freely downloadable from the link indicated on this slide. Inside the MRI scanner subject is shown a bunch of video clips, in this case from BBC Planet Earth, of underwater life, and these video clips are interleaved with resting blocks. So during this whole period, which is about 6 to 8 minutes that the subject is inside the scanner, the functional scans are sent about every 3 seconds to the external PC where we do network analysis. We're trying to get a characterized brain activity pattern and then map that to the BrainMap database. And the brain activity is then described in terms of 50 behavioral profiles. And the one we expect, or the set of behavioral profiles we expect to see activated most would be vision, because it's related to the experimental task design. To prepare for the experiment, we have a first step prior to real-time acquisition, where we acquire an anatomical scan and a functional scan of the subject. This takes about 10 minutes. During the second step, we open up the Neu3CA-RT program, which is written in MATLAB, and we locate the specific files that we just acquired for further processing. We also set up the correct directories for the Neu3CA-RT program. For pre-processing, everything is mapped to the functional space, which is the space in which network analysis occurs in real time. The anatomical scan is co-registered and segmented, and our network model that we use for network analysis is mapped to our functional space, and everything then is resliced to the functional resolution. Here you can see that being executed, and the process has just been sped up for the purpose of this video. The specific fMRI network model that we use can be viewed in these pictures. We have a set of 20 images that together form the model for our data. Next comes the real-time initialization, just before real-time processing, which is very quick. We initialize the GLM design matrix, we initialize the task design, and we start the scanner remotely, and then we press the button to start the experiment. Finally, we have the real-time processing, which runs about 8 minutes in total and occurs every 3 seconds when each dynamic functional scan comes into the external PC. First, we convert the data format to NIFTY. Then we realign each image to the initial functional scan that we acquired. We run our spatial GLM and remove any trends that would confound the data. And then we map our characterized activity to the BrainMap behavioral space i.e. in terms of 50 behavioral profiles. Here you can see that being done on the program. The bottom left figure shows each dynamic functional scan as it is acquired. On top of that, we have the task time course, where you can see the black strips indicating the rest period for the subject, and the white strips indicating the period during which the subject receives a visual stimulus in the form of the video clip. On the right-hand side, for each dynamic, we display the correlation values of each of the 50 behavioral profiles from the brain map database with our current spatial activity map from the functional scans. These values are shown over time on the top left figure, specifically for the visual behavioral profiles that we are interested in. The color scale on this top left figure is blue for strong negative correlation and yellow for strong positive correlation. So we expect to see yellow bars right on top of the white bars of the task time course, which means that there is high visual activity in the brain during the period that the subject views these video clips. And we also expect to see blue bars right on top of the black parts of the task time course, indicating lower visual activity during the rest periods. As we progress to the end of the experiment, one can see the expected interleaved yellow and blue bars in the visual behavioral time course, although the data is still a bit noisy. This provides us with a basis for further work on the Neu3CA-RT program.